All right. Thanks for joining us today. My name is Chris Stepanuk. I am a extension leader and associate director for Lake Champlain Sea Grant, and happy to be here to facilitate today's fall research seminar with Tim Mihuc, Dr. Tim Mihuc from SUNY Plattsburgh, just to give you some guidance about how we run this because we're using a Zoom webinar. You have access to the questions and answer opportunity at the bottom of your screen. You can type a question in or let us know that you have a question there. And then after the presentation, we'll let people turn on microphones or just use the typing in function for the questions and answers to let Tim answer live. If you need help on Zoom, I'll drop this in the chat after I get finished giving you this explanation to a link to help you with Zoom. And just so everybody knows, we are recording this webinar. Today is the second of 4 seminars for this semester, again with Dr. Tim Mihuc from SUNY Plattsburgh. But just to mark your calendars, our November and December webinars are off cycle due to the holidays. So mark your calendars for November 13th and December 11th from 12 to 1 to hear from other researchers from the Lake Champlain Basin. And then today we have Tim Mihuc, Associate Professor of Environmental Science at SUNY Plattsburgh, and he coordinates the Lake Champlain Research Institute. He is also our Associate Director for Lake Champlain Sea Grant, again, based at SUNY Plattsburgh. So I will stop sharing, and I'm going to turn it over to Tim.
Hi, folks. I'm going to share my screen now. Make sure everyone can see that okay.
It's good, Tim.
All right. And so I'm going to do this a little different than a traditional research seminar. Um, our lab at LCRI has done a series of short stories, mainly for children, and I'm going to follow that story for some of this dynamics of invasions into Lake Champlain. Before I get there, I want to just do a little introduction to Lake Champlain in general and plankton in general. And after we go through the story, we'll cover a few other things and talk about some of the major invasions in the lake, particularly to the main lake of Lake Champlain, the planktonic main lake, and what the implications are of those species invasions. So we all know Lake Champlain pretty well. This is a good map of it. This is the long-term monitoring map, which shows the sites for all the river sampling and all the lake sampling, which are those numbers and dots, for where the monitoring essentially is happening for LTM. And what I'm going to use a lot of for the data is the long-term monitoring data to illustrate the dynamics of what happened when zebra mussel invaded and fishhook waterflea and spiny waterflea to some extent we'll talk a little bit about today. Before I do that, I want to just introduce the concept of plankton, in particular zooplankton. But as you may or may not know, the lake water, the main lake itself, is full of a planktonic biological community that's very important, particularly for production of the fishery. So we have phytoplankton or algal blooms, which are fed on by this whole group of animals called zooplankton. Sorry, my slides seem to be transitioning. I'm going to go back to them as I move on. And those are 3 groups: Cladocera, copepods, and rotifers. So that's the zooplanktonic community that really drives the food web that supports the fishery. There's really been 4 main invasions into the main lake of Lake Champlain in the last 30 plus years. The zebra mussel in the early 90s, the alewife in the mid 2000s, and the spiny waterflea in 2014, and then finally the fishhook waterflea in 2018. Most of the story you're going to see next is about the early invasion of the zebra mussel into the lake associated, and then the leading up to the invasion of the fishhook waterflea and spiny waterflea. So what I'm going to do is just go through this story that we wrote, my daughter, Sarah, and myself, with some help from a wonderful undergrad student who illustrated this, and then edited by Evelyn McCord. So I'm going to just go through and read through this story pretty quickly. So I'm going to move my, hold on a second. There we go. So after their previous defeated attempt, the pirates' Waterflea were not ready to give up their quest to invade Lake Champlain. They retreated to a safe place, pulled up for the winter as resting eggs, and prepared for another invasion attempt the next year. Awaking from a long winter's resting egg, Captain Fishhook gave the order for Spiny Beard's revenge. Spiny, as Captain Fishhook had come to affectionately refer to his first mate. I have a plan, matey. We are going to get you in first, and then I'll come with reinforcements later. Spiny chuckled. Excellent plan, Captain. I'm ready and able on your orders, sir. Captain Fishhook then described in great detail the plan for Spinybeard by hook or crook. He was to invade Lake Champlain from the south using a surprise strategy, a full-scale invasion. First, Spiny had to find a nearby lake from which to stage his dastardly plan. He bid farewell to Captain Fishhook and started his journey to Lake Champlain. On the southern route, he tried several of the Finger Lakes in central New York, only to realize his miscalculation. None of these blasted Finger Lakes are directly connected to Lake Champlain. He got as far as the last Finger Lake, and he was just about to give up when he realized the most amazing thing. Lakes are connected by rivers. Sometimes, even when a natural river connection does not exist between two lakes, someone makes one for you. Spiny needed to find the perfect connection to Lake Champlain, so when he went to see an old friend, Stripes the zebra mussel. On the right, you see a quick fact box about zebra mussels. Invaded the lake in the 90s. Stripes was pretty good at invading lots of ecosystems. Spiny knew from his time training on Lake Ontario's pirate invasion fleet. He also knew Stripes had already invaded all of the Great Lakes, the Chicago and Illinois River, the Mississippi River, all the way down to the Atchafalaya Swamp in Louisiana, and most of the rest of eastern North America. As Spiny says, no one does it better than Stripes. No one. If anyone knew how to stage an invasion in Lake Champlain, it would be Stripes. Said Spiny as he knocked on Stripes' door. Are ye home? Stripes got up from a plate of nice, tasty rotifers to answer the door. Well, hello there, my good friend. Spiny, it's been a long time, said Stripes in his thick British accent. How may I be of assistance? Spiny replied, I need information and lots of it. I'm planning to invade Lake Champlain, and I need your help. Help, screamed Stripes. You don't need my help. Invading Lake Champlain is easy. Just use the bloody canal. Spiny thought, wow, we really just use boats. Stripes replied, what's a blasted canal? Stripes replied. Spiny, you numbnut, a canal is a human-made artificial river like a highway humans use to move around where rivers do not naturally exist. You blimey fool, you need to use the Southern Canal to access Lake Champlain. I'll give you directions on how to get there. Spiny replied, okay, thanks, matey. Stripes continued, look, this is tricky. But once you know where they made canals, you will make it there easily. You can get to Lake Champlain by going up the Hudson River to the Champlain Canal, then go north, and after a few miles, you're in the South Lake. From there, the whole lake awaits. Spiny said, I can't swim upstream in the Hudson. I'm a plankton. That's not my niche. Stripes replied, true. Okay, we need a more complex plan. First, you will need to invade a lake nearby with a river connection to the canal. That way, it's all downstream from there. You can visit me friend who apparently is in the Hudson Valley, Harry Hydrilla. He will know the way. To the right is the zebra mussel invasion pathway through Lake Champlain from the south through the Champlain Canal, most likely, and then the invasion in subsequent years in the 90s throughout all of Lake Champlain. Let's stop for a minute and talk a little about some data. This shows the response of rotifers and mysids in Lake Champlain, two of the components of the planktonic community, with a dramatic crash in population numbers associated with the zebra mussel into the lake. We'll talk a little bit more about this later. Just to get a little more into rotifers, this shows the community dynamics of the different taxa of rotifers, their different population numbers, and how they all declined in the early 90s in the main lake, associated with the zebra mussel invasion. Also, you can see that the rotifer that is common after the invasion, this one with the fish scale looking pattern, is called Conachilus. It's a gelatinous colonial rotifer, and zebra mussels cannot filter them because they live in a colony. So they have taken over the rotifer community in the lake after zebra mussel invasion. All right, back to the book. Spiny turned to leave and said, "Thanks, matey. Eat lots of those rotifers. I hate those little scallywags, especially the ones in Lake Champlain." Stripes replied, "Long live and may your journey be full of invasion success." Spiny made his way to the Hudson, home of Harry Hydrilla, to find out more about the rivers and canals flowing into Lake Champlain. Upon arriving at a pond to meet Harry, Spiny said, "Hi, Harry. Stripes sent me to see you. My, what a thick mat you have. That would smother a native fish for sure." Harry said, "Why, yes, it will. Some may think I'm good when I invade, but I assure you it's bad for the natives. Makes them restless." On the right, you see a fact box about Hydrilla verticillata, an invasive plant from South America throughout most all the rest of North America. Spiny then continued his quest by asking, "Harry, I don't have much time. How do I invade Lake Champlain?" Harry stopped, pondered the situation, and said, "Okay, you need to hitch a ride on a boat. Pick one that's traveling toward Lake Champlain. Maybe you get into a body of water connected to Lake Champlain by a river or a canal. Then you can carry out your dastardly revenge and invade from there. I suggest Lake George, or maybe Sacandaga Lake. Fastest way to get to Lake Champlain would be Lake George, but they have strict boat cleaning regulations. It'd be hard for you to sneak in, so you may have to get to Sacandaga first, and then go from there." "Okay," said Spiny. "Do you know a boat I can ride on?" Harry replied, "Try one of those eastbound boats from Lake Ontario. Lots of those people fish in Sacandaga Lake in particular, and many of them don't clean their boats." "Excellent plan. I'll get to Sacandaga Lake and invade from there." Watching Spiny slink away, Harry thought about his own secret plan to invade the Adirondacks. He wasn't happy staying in the South. His plan was to continue his expansion north, first Lake Champlain, then the world. Since hitching rides on unwashed boats was something Spiny had lots of experience doing, he knew exactly what to do. He liked being a dirty pirate. He jumped on the first passing boat. He ended up right where Harry said he would be, in the beautiful Sacandaga Lake. From there, he slinked his way down the Sacandaga River until he entered the mighty Hudson River. From the Hudson, Spiny found the entrance to the feeder canal for the Champlain Canal, just as Harry had predicted. He had found his destiny, and finally, after a few attempts, he made the rest of the trip through the canal, then into Lake Champlain. Just for fun, he invaded several other lakes nearby, including beautiful Lake George, where he exclaimed, "What a pretty sheet of water!" The diagram to the right describes the timing of the invasion dynamics of spiny waterflea into Great Sacandaga Lake in 2008 and a subsequent series of other lakes between 2008 and 2014 when it was found in the canal 1 lock away from Lake Champlain. Finally, with dreadful intentions, he made it to Lake Champlain, his ultimate destination, where he said, "Here I am, our mateys. Time to eat some native plankton and make this ecosystem mine." The spiny waterflea population grew and grew until there were so many waterfleas and the local anglers cried, "Enough! These darn spiny waterfleas are clogging all our lines and destroying the food web in our lake. The fishing here is ruined." And to the right, we see some of the early samples found in Lake Champlain in 2014. The top one is a female spiny waterflea with a brood of 4 on its back, a brood of eggs. And the bottom one is one of the early samples with several hundred spiny waterflea specimens, literally within weeks of the first specimen collected in the lake. While Lake Champlain was a very hospitable place for Spiny and his mates, eventually he became restless and developed a longing to explore new waters. As he developed plans to expand beyond Lake Champlain and into other lakes, he shouted, "Where will I go next? Only the future will tell. Watch out. I may be coming to your lake. I hear the Adirondacks are a very nice place. Arr! Has anyone seen a boat leaving Lake Champlain that I can hitch a ride on?" So this shows, again, that more broadened out invasion map from the Great Lakes to Sacandaga Lake, a series of Adirondack lakes. By 2012, the spiny water flea had invaded Lake George, the Champlain Canal, and a number of other Adirondack lakes. And in 2014, successfully invaded into Lake Champlain. That's where the story used to end. But this last little bit is a result of the invasion of Captain Fishhook himself. So a few years later, Captain Fishhook made many attempts to invade Lake Champlain and was very motivated after Spiny Beard was successful. Finally, after years of trying to catch a boat headed from Lake Ontario, in 2018, our beloved captain exclaimed, "Argue dastardly cowards, I've made it to Lake Champlain," as he jumped off a fishing boat that had just returned from a trip to Ontario. This unknowing and currently unknown boat operator has inadvertently become infamous in the lore of Lake Champlain's planktonic community. And that's pretty much the end of that story. The theme of the story is obviously watch out for invasive species. Let's try to prevent invasive species from being transported. Probably the other main theme of that story is what transports invasive species? We do. Thanks to the people who contributed to this, like I said, Sarah Mihuc, Evelyn McCord, and in particular to the wonderful animations done by a student here at SUNY Plattsburgh, Caitlin Merzaka. So what I want to do now is just get into a little more on the details of this. This is from a paper that's published a few years ago by one of our grad students, Zach Cutter. It shows the two invasion maps of both of those species. So you see the 2014 map on the left, showing that in early August, we detected virtually no spiny water flea in the lake. I'll point out that these samples were taken for 4 years prior to this at all of the lake monitoring sites, and we detected no spiny water flea in any of those samples for over 4 years. We finally detected some. Within 2 weeks, they'd expanded to much of the lake, and by 4 weeks, they'd expanded lake-wide in extremely high densities. And many lakes, we don't have this data because we're not sampling before the invader invades. We have the same data from 2018. Again, no fishhook water flea detected for years and years and years before early August 2018, and then a few detected in mid-August at several sites. Within 2 weeks, they had expanded throughout the entire lake. So one of the questions is, how did they do that? Which is interesting. This is again the review of the spiny water flea invasion map. This one of the early samples we detected. This is the fishhook water flea. Previous invasion was only known from the Finger Lakes or Lake Ontario. So it came to Lake Champlain from one of those two sources. We're hypothesizing based on the story that it's most likely Lake Ontario. So one of the points about these species is they're members of the Cladocera, which are the water flea's common name. And these are some of the native cladocerans that live in Lake Champlain. These are small plankton that feed on algae, but they have a very interesting biology, which explains how they could expand so rapidly in the lake. They're parthenogenetic. For those of you who don't know what that means, it's essentially called virgin birth, which means females can produce clones of themselves during summer conditions when they need to reproduce quickly. And they bypass normal sexual reproduction and just produce clones. And they can do that very quickly. So the spiny water flea is parthenogenetic. So once it gets in, viable females can just clone themselves very quickly and expand the population very quickly to respond to either the invasion or to changes in a food source, for instance. So an interesting biology there among all the Cladocera, which is their ability to be parthenogenetic. Another major component of the plankton are the copepods. And so these are some of the common copepods in Lake Champlain. These are larger, they grow slower, they don't have the ability to be parthenogenetic. They're larger, they feed on larger prey items and larger food items for the most part. Many are omnivorous, both predators and feeding on algae. And then the last common group, which is discussed a little in the story, is the rotifers. And these are the ones that really declined a lot in Lake Champlain with zebra mussel invasion. They're very small, they don't swim well, they're a lot like particles, probably how they get filtered out by mussels. They get directly filtered like all other particles. They don't, again, they're parthenogenetic as well, just like Cladocera, but they're also much smaller, with much poorer swimming abilities. Some of this information you can find in a few other works that we have, published works out on various dynamics of the invasions or long-term patterns in Lake Champlain. I'll review a few of those really quickly here. This is a paper recently out by Zach Cutter, one of our master students who still currently works for the Research Institute, on the dynamics of the plankton community and how it changed with the invasion of both fishhook and spiny waterflea. So this is really based on the long-term monitoring data, understanding that dynamic. You see one graph over here, which is Daphnia retrocurva, a very common cladoceran in Lake Champlain. And I won't summarize that graph to a large extent other than to point out whenever fishhook and spiny densities are high, Daphnia retrocurva densities are always very low in the lake. We have done some other work on invasion dynamics associated with the zebra mussel. In this case, we ran a complex evolutionary model called hybrid evolutionary algorithms with a colleague of mine who's actually from Australia. And this model, essentially, it's pretty complicated, but it showed that the decline in rotifers was related to the pattern in zebra mussels in invading the lake, and also some linkages to a few other taxa in the planktonic community, in particular the mysid shrimp. I had shown you that graph where mysids and rotifers both declined upon invasion of zebra mussels. So we know these invaders into the planktonic community have had some impacts. Both zebra mussels and fishhook and spiny waterflea have all had an impact on the planktonic community of Lake Champlain. And I'll review that really quickly on back to the original invader, the zebra mussel. This shows the decline in rotifers in gray, the zebra mussel expansion in blue. They're directly correlated. As zebra mussels came in and became abundant, rotifers went away in the lake. And then you can see some patterns going out long term. Zebra mussels are widely known in the literature to be able to directly filter rotifers in the Great Lakes and in other ecosystems, and very likely are, having that direct influence on Lake Champlain's rotifers to the demise of a whole part of the planktonic community in the lake. And again, this graph was also briefly in the story. It shows the decline in rotifers and the decline in mysid shrimp. They're at the top end of that planktonic food web, so we didn't expect to see this decline. But there's a paper out by colleagues at UVM and a student, Suzanne Ball. We have a paper out on this decline in mysids in the lake in the 90s. So rotifers really took it on the chin when zebra mussels came in, except for Conachilus, that gelatinous colonial rotifer. All the other species, these all cool looking things around the edge, Calicodia, Keratella, Polyarthra, they all declined dramatically in abundance. We were not actually detecting them in the samples for years. We currently do detect them now, but in low abundances in the lake, but it took 10 years or so for many of these species to even recover to be detected in samples again. And I want to just finish with a summary of just generally sort of how this all puts together in the food web context. I know I'm talking about a lot of small organisms here that most people don't know that much about, but we have the zebra mussel that's come in, definitely had an influence on the food web on the rotifer side. These orange boxes are the plankton, the zooplankton, and then in the green boxes are the algae or phytoplankton. It's kind of a hypothetical food web for Lake Champlain. Mysis dulumiana is here. It's a large planktonic predator. Leptodora is another one that's native in the lake. We have all these invaders that have come in and had various effects from top down or bottom up on the food web, including alewife, which we haven't really discussed a lot here today, but spiny waterflea, fishhook waterflea, and zebra mussels affecting different parts of the food web. And I'm going to summarize that just on the zooplankton here real quickly, with zebra mussels having this influence on rotifers and mysid shrimp, and then spiny waterflea, and then the most recent invader, fishhook waterflea, having an effect more on cladocera and copepods, and they feed on different size organisms, so some differential effects there too between spiny and fishhook waterflea on the plankton. All of this ends up affecting our native planktonic communities, including declines in the native Leptodora, potential impacts on Mysis diluviana, a very important food web component in the lake. The forage fish are feeding largely on mysids, and that supports the trout fishery in the lake itself. I want to just highlight a few projects ongoing, some master's students who have either finished their work or are doing their work now in LCRI at SUNY Plattsburgh. And one of them, Marshall at the bottom there, has his thesis defense on vertical migration of plankton and relationships to both fishhook and spiny waterflea invasion on December 4th at noon at SUNY Plattsburgh. The others are some thesis work on mysids, the paper that I mentioned on fishhook and spiny waterflea, and then we've looked at long-term patterns in phytoplankton as well throughout the lake system. And I used to end my talk about Lake Champlain plankton with this slide about what the future potential invasive zooplankton would be. It's a little sad and unfortunate that I now have to use this slide to basically point out that both of these have invaded the lake and have had an impact on the planktonic community. And I want to finish with sort of just a little bit of advocacy here. Why should you care about plankton? These tiny little creatures in the food web that you may or may not have even known existed prior to me talking about them today. So I'm going to tell you a little story about Flathead Lake, Montana. And this comes from a grad student I had a few years ago who ended her thesis defense with this argument. And I've used it ever since because I think it's really useful. So in Flathead Lake, you have this great planktonic community that supports a salmon fishery, kokanee salmon in particular. It also supports a pretty strong grizzly bear community and our bald eagles are the symbol of our nation. Flathead Lake's a wonderful, beautiful place to visit. And in Flathead Lake, all of that stuff is important. It's important to the people there and the communities and those who manage the community. And what they had was an invader. In this case, the invader story is the mysid shrimp, a possum shrimp. They were deliberately put in Flathead Lake by the managers who said, this will help our lake. It will feed the fish. So this was a deliberate introduction. It was not random. It was not on a boat or anything else. It was managers who said, we will introduce mysids to Flathead Lake. When they did that, the plankton community crashed. Mysids ate all the plankton as a top predator. That had influences on kokanee salmon and the other salmonid populations. The kokanee salmon crashed 2 years after they introduced mysids to the lake, which had influences on lake trout. And then eventually grizzly bears and bald eagles didn't have as much food, so they moved on to other places. So the moral of this story is, if you don't care about zooplankton, you don't care about America. Thank you, and I'm happy to take any questions you might have.